Hello and welcome everyone to my YouTube channel once again. Today the topic of discussion will be immunoglobulin G. Let's proceed. Immunoglobulin G constitutes about 75 to 80% of total immunoglobulins in our circulation. So it is a major antibody. It's the chief serum immunoglobulin. Half-life of immunoglobulin G is about 23 to 25 days. It is a monomeric antibody. It has two heavy chains and two light chains. One immunoglobulin G molecule can bind to two antigens. Immunoglobulin G subclasses. So immunoglobulin G has four subclasses, namely immunoglobulin G1, G2, G3 and G4 due to gamma 1, gamma 2, gamma 3 and gamma 4 heavy chains respectively. So this is a picture or a diagram of different immunoglobulin G's subclasses. As you can see here, the yellow portion of the immunoglobulin indicates the light chains and the green ones are the heavy chains. You can see one particular thing in these immunoglobulins, and that is the uh, length of the hinge region. So this is the hinge region of each immunoglobulin and the length is varying in each subclass. Not only is the length of the hinge region is different, but there are subtle differences in this FC region of each immunoglobulin subclass, because of which they have different functions. So let's see what are the immunoglobulin G subclass functions. We just saw the different immunoglobulin classes of IgG. Now we will take a look at their functions. Immunoglobulin G1, G3, G4 cross the placenta and protect the fetus. Immunoglobulin G3 activates complement system. Immunoglobulin G1 and G3 bind to the Fc receptor on phagocytic cells, monocytes and macrophages and mediate opsonization. So the three major functions that different immunoglobulin classes have is to protect the fetus by crossing the placental barrier, by activating the complement system and by mediating opsonization. So have you ever thought which is that immunoglobulin which provides us safety or immunity when we are newborn and our immune system is not fully developed? Firstly, this kind of immunity is known as natural passive immunity, and immunoglobulin G provides us natural passive immunity when we are newborn. It is the only maternal immunoglobulin that is transported across the placental barrier, and this is the maternal immunoglobulin which is transported in the fetus from through the placenta. So this is the maternal immunoglobulin and it is seen in the neonatal circulation for 2 to 4 months. So once the child is newborn, this immunoglobulin remains in the circulation for 2 to 4 months, protecting the child with natural passive immunity. Now we come to one more property of immunoglobulin G and one more phenomenon which is known as isoimmunization, and this RH isoimmunization is known by another name which must be familiar to you, and that is RH incompatibility. So what is this RH incompatibility? Now when as you know that the immunoglobulin G can cross the placental barrier. So this phenomenon causes RH isoimmunization. This occurs when the mother is RH negative and the fetus is RH positive. In the first pregnancy, the immunoglobulin from the fetus enters the mother during childbirth, and this leads to formation of Rh antibodies in the mother. So these antibodies are formed against the Rh positive blood group and they stay in the mother's circulation. Now in the second pregnancy or in the next pregnancy, when the when the fetus is Rh positive, then these Rh antibodies enter into the fetal circulation, causing fetal hemolysis, neonatal jaundice, and neonatal death or miscarriage. How can we protect the second pregnancy in such a case? So now, immediately after uh, the delivery of the first child, if there is passive administration of RHD immunization given to the woman, then these antibody secretion will against the RH group will be inhibited, and this is how the second pregnancy can be protected. So this is known as RHD immunization. Now, immunoglobulin G is known as a late antibody. It is known as a late antibody because it appears after initial immune response by immunoglobulin M. It is the antibody that is seen in secondary immune response. Let us understand this better with the immune response curve. So this is the immune response curve. This is the antigen when when the human body is exposed to this antigen for the first time. Immunoglobulin M comes and protects, and gradually the immunoglobulin G rises in the subsequent attacks. So that is why immunoglobulin G is known as the late antibody, whereas immunoglobulin M is known as the early antibody. So, opsonization effect of immunoglobulin G and antibody dependent cell mediated cytotoxicity. What are these phenomena? Let us try to understand. Opsonization effect is shown by immunoglobulin G, and it is a very important immune response provided by it. You can see that this is a natural killer cell. Now this immunoglobulin G is binding with the natural killer cell through the Fc portion. Immunoglobulin G can recognize the target cell. This has recognized the target cell which has to be killed. So the pathogen is now flagged by immunoglobulin G for phagocytosis, and now it is phagocytosed. This is one process of opsonization. Now we will take a look at antibody dependent cell mediated cytotoxicity. Here this is a large pathogen which has been recognized by immunoglobulin G, and now immunoglobulin G has completely opsonized it, which means it has formed an envelope around it, it has covered it. Once this is done, the natural killer cell is stimulated to produce release certain toxic granule contents, and these toxic granule contents cause death of the pathogen by membrane damage and necrosis. And this is what is antibody dependent cell mediated cytotoxicity, because immunoglobulin G enhances this opsonization process and killing of the pathogen. These are my references for the lecture. Thank you for watching. Like, comment and share, and subscribe to my channel.